All right, we are on chapter 6, section 4, and I have to say, this is one of my favorite sections. And I know it's really short compared to the others, but it's really awesome. All right, so I hope you love actin as much as I do. So we're going to talk about the third type of protein from this chapter, and that is a structural protein. I know your slide here says structural proteins, the actin, myosin, motor, but really actin is structural, whereas myosin is a motor. Myosin is not really a structural protein. But all right, so for a really long time, people really wanted to know how you can have a muscular force, right, that can be exerted at the cellular and molecular level. And so this model was first proposed in 1954 from two different groups, one at the University of Cambridge in England and one at MIT in Massachusetts. And so it was called the sliding filament model. So it stated that muscle cells have these filaments, right, that have two proteins in them majorly. I mean, there's more, but the two major proteins are actin and myosin. And so here's our pretend, this is a very simplistic drawing. This is our myosin filament, and this is our actin filament. And so they said that this sliding filament theory, that there are two proteins, and the actin and the myosin slide over one another during muscle contraction, and they use chemical energy in the form of ATP in order to make this happen. And so before we can really talk about the super details of this kind of a function, we kind of need to do a background on the arrangement of proteins inside of a muscle cell. So I don't know how much background you guys have, so I'm kind of gonna give you quite a bit of background. Muscle cells are kind of unique because they're not individual cells. They are actually fused cells. So you have one cell that fuses to another cell that fuses to another cell. And we call those fused cells myoblasts. So myoblasts. And so within one myoblast, you're going to have many nuclei. And so these myoblasts are cells that are covered in a plasma membrane, just like normal cells, but we call this plasma membrane something special. We call it the sarcolemma. So it's still a plasma membrane, but we call it something special because it's muscle. Okay. And so inside of these muscle cells, you have these like basically strands of bundles of fibers, and we call these strands myofibrils. And the myofibrils are composed of myosin and actin. And so whenever we say that myosin is a single protein, but myosin doesn't exist in the cell usually as a single protein, usually, at least in muscle cells, it basically hooks on multiple in a row. And so we call that filament, that row of myosin molecules, we call them thick filaments. And then actin is a globular protein. And the actin will actually, you'll get, you know, one actin molecule hooked to another actin molecule, hooked to another, hooked to another. And so it turns into a polymer. And so that actin polymer is then called a thin filament. And the thick and thin filaments, right, if this is your actin and here's your myosin, they overlap each other. So we'll see that in some pictures, and I'll show you kind of how that's structured. Okay, so this is an example of one myoblast, right? And you can see that it is covered, let's see, in a sarcolemma, right? So this sarcolemma wraps the entire cell. You know, it goes all the way over here, all the way over here. So it's just the plasma membrane that wraps around that cell. And then, right, inside the cell, we have nuclei. So here's one nuclei, and the nuclei kind of get shoved to the edge of the cell because so much of the cell is filled up with all these bundles and bundles and bundles of what are called myofibrils. So here is an example of one myofibril. Here's another myofibril, and another, and another, and another, and another. And then here's a last one that they basically just kind of pulled out. right? And so these bundles, these myofibrils, are composed of polymers of actin, which we call thin filaments, and polymers of myosin, which we call thick filaments. And so this repeating unit of actin and myosin are called the sarcomere. So if we looked at one myofibril and we took a little piece of that one myofibril and kind of zoomed in on it, what would we see? Well, see if I can zoom in, right? We would see in the dark red, and I'll kind of highlight it here, in the dark red, this is our thick filament, this is our myosin, right? And then we also see our thin filament right here, this is actin. So you see how in this area, right, they overlap. So we have overlapping here and we have overlapping here, all right? And so all of these filaments, let me see if I can erase some of this, are hooked to a set of proteins that look like a zigzag. And so they call this set of proteins Z-disc proteins because it kind of looks like a Z, right? And so from one Z-disc to another Z-disc, we call that a sarcomere. So a sarcomere is composed of actin and myosin in kind of an overlapping function, in overlapping fashion, excuse me. And then many sarcomeres lined up end to end. So if this is one sarcomere, here's another sarcomere, here's another sarcomere, here's another sarcomere, that's going to turn into one long myofibril. Okay, so our sarcomere runs from Z-disc to Z-disc, and then during muscle contraction, what is going to happen is the distance from Z-disc to Z-disc is going to shorten. So the two Z-discs are going to get pulled in towards one another. And it's a small change. It's a small change. But all of the sarcomeres in the entire myofibril, in the entire myoblast, and like in the whole muscle itself, are all going to contract at the same time. And so it's going to end up being something big, something strong, because all these little things are happening at exactly the same time. And so in order to pull those Z-discs in, you have to have an anchor, right? If it's the myosin that's pulling on the actin. And so if you're going to pull, then that actin has to be hooked to something. Well, what's the actin hooked to? The actin is hooked to the Z-disc, and that's why the Z-disc will come closer and closer to one another. All right, so let's kind of review some terms. So myosin, this is what makes up our thick filaments. And so they're arranged to where what's called tails are associated in the middle of the sarcomere. So if you have a myosin filament, it's going to kind of look like this. It has a head region and it has a tail region, right? And they're actually usually two kind of hooked together. They kind of wind around each other. And so you actually end up with two heads instead of one. But what ends up happening is it's not just two. This happens again and again and again. And so you have a head group and another head group. And I'm really not an artist, but what you can see is that the tails kind of line up and then the heads are stacked one after another. And so you end up with the middle section of a sarcomere, right? Because the ends of the sarcomere, the Z-discs, are basically over here, right? And then you could have another one like way over here. And so what you have then attached to the Z-disc are actin filaments, and they actually are a DNA helix, right? So you have one, and then you have another helix that's going to come this way, right? Okay, and so those head groups on the myosin, so this is our actin, and that's our thin filament, right? And so the globular heads are at the ends, and they can actually bind onto the actin and pull on the actin. So the actin filaments self-assemble, and they stay in these long polymers. And they're not the only protein that are in those thin filaments. There's another protein, And that protein is called tropomyosin. And the tropomyosin kind of weaves and wraps around the actin filament. And it actually, in the resting state, covers the myosin binding sites on actin to prevent myosin from binding when our muscles are relaxed. And then what's going to happen is there's a third set of proteins, right? And they kind of sit in repetitive spots along the actin filament. And these are called troponin complexes. And so it is actually, when calcium comes in, it's going to bind to the troponin complex. And the troponin, and I'm going to show you this, so you don't have to write all this down, just hang tight. The troponin complex is going to bind to the calcium and undergo a conformational change. That conformational change is going to make our tropomyosin that's wrapped around the actin undergo a conformational change. And then you'll have sites on the actin that are now exposed, and the myosin can now bind to the actin and then undergo pulling. So let's look at kind of how that looks. And you would have, and what I didn't draw here is, right, that you have myosin going this way as well, right? And so this is what one sarcomere basically looks like. And then you also have, and I don't want to spend too much time, but drawing, you have actin coming out this way, right, as well. So that it's the 2Z discs coming in that way. All right, so here's kind of a better drawing, but I kind of like drawing it myself because then you can put it in piece by piece, right? Okay, so here's our Z disc, and what's unfortunate is that they're not showing this as really a Z, but it is really a Z. So you have your actin, which is hooked to your Z disc, right? And you've got these troponin complexes, right, that occur every so often on the actin filament, right? And then you have, on top of that actin filament, here is your tropomyosin. So here's your tropomyosin. If you can see it kind of cover those binding sites, and it covers it on the other strand of actin as well. So it kind of wraps around. So then every so often you can see where that troponin complex fits in kind of the divot right there, okay? All right, so you have your myosin laid out in the middle. Your myosin is your dark red stuff right here, right? And it wants to bind onto that actin, but it can't because the troponin complex doesn't have calcium, and so the tropomyosin is blocking those sites, right? And so if you look, right, there's going to be an area where it's going to be really dark if we look at this under the electron microscope, where we have an overlap of this thick and thin filament. It's going to be very dark. Then you're going to have a portion that's going to be light because we only have actin and this other protein called titin. And titin is like a spring. They draw it kind of like a spring here so that it can stretch and compress depending on the position of myosin. And we're gonna look at how that works. So it's really the calcium binding to the troponin complex that is going to really regulate contraction. So it's calcium binding to troponin, TROPO complex, that regulates muscle contraction, right? Because that's where it starts. Calcium comes in and it binds to troponin. Troponin induces a conformational change in the tropomyosin, right? So then you get a conformational change in troponin. That causes a conformational change in tropomyosin. And this exposes myosin binding sites on actin. Binding sites on actin. Okay, and so that's what it allows. So if you have no calcium, right, the troponin and the tropomyosin are hiding those actin binding sites, and myosin can't see them. And so you're not going to have any contraction. But when you get calcium, everything undergoes a conformational change, and now myosin can see it. So let's see kind of how this looks. So this is your electron micrograph, right? This is electron micrograph. And they're only showing it, they really should show an electron micrograph of both relaxed and contracted, but they're only showing one. But here in B, you can see both. Okay, so B is the relaxed state. Okay, you can see the distance from Z disk to Z disk is relatively long. In the next one, this is our contracted state. So the distance from Z disk to Z disk has been shortened. So that's how you know the relaxed state from the contracted state. You can look at the distance from Z disk to Z disk. So in muscle contraction, what's going to be initiated, what's going to cause that initial change, is you're going to get a neuronal signal, right? So a neuron is going to stimulate a muscle cell, and that's going to result in release of calcium. So you get nerve cell, nerve signal, right? And then you release calcium from, y'all have heard this before, the sarcoplasmic reticulum, the reticulum, right? So the sarcoplasmic reticulum has lots and lots and lots and lots and lots of calcium. And so it's going to basically dump calcium into the cytoplasm. The calcium, right, is going to bind to troponin, right? The troponin is going to undergo a conformational change. Then tropomyosin is going to undergo a conformational change. Now those myosin head groups can attach to the actin, and the myosin head groups are going to act as a ratchet, right? They're going to, it's like, this is our myosin, I'm sorry, this is our actin. Our myosin's basically going to grab onto it and ratchet it. And so it's going to pull it in one direction. Right? So this is going to take a couple of different things, right? It's going to take an influx of calcium, and it's also going to take some ATP to make it happen. And we haven't quite talked about that ATP need, right? But here it is, right? You have to have ATP and you have to have calcium in order to have muscle contraction. So when we look at this, what's important to note, right, is that there are different, when we're looking at an electron micrograph, how do we know contraction has happened? Well, we measure the distance for a couple of different bands here. We're going to look at this middle band, which is called the A band. Then we're going to look at, on either side between Z disk and A band, is something we call the I band. Why they couldn't name it something more accurate to what's in it, I don't know, but they didn't. So the A band is very, very dark. So if you look in this electron micrograph, this is our A band right here. And then we have this light thing right here, and we call this and this the I bands. And so on either side of the I bands, what you're going to see, if I can pick another color I haven't used yet. Here we go. Let's do green, right? This is the Z disk. Here's another Z disk, right? It's that dark black. And so when we look at this, what we're actually going to see is that when the myosin hooks onto the actin and basically ratchets it and pulls it in, the A band is going to stay the same. The A band distance is going to stay the same. But when the actin gets pulled, what's going to happen is this I band is going to shorten, because as it's being pulled, you have more actin overlapping the myosin. So you have less actin all by itself here. Well, look how much actin you had all by itself here, a much bigger space. And so this is how muscle contraction works. You're shortening just the I band on either side, and all of these sarcomeres happen at the same time. They all contract at the exact same time to result in your full muscle contraction. So let's look a little bit more detail in how that myosin ratcheting actually works. And so the first thing you have to know is how's the myosin going to actually attach to the actin. So this is in the presence of calcium, right? So all of this stuff is happening in the presence of calcium. So you have, here's our troponin complex, right? Right about here. We have another one, and we have another one. That's our troponin complex. And the troponin complex is going to bind onto our calcium, right? And so what you can see is while it's unbound, this is an example of an actin binding site for myosin. So myosin is supposed to bind right here, but what's going through it? What's hiding it? What's covering it? That's the troponin. So when our troponin, I'm sorry, it's our tropomyosin. What did I say? Tropomyosin. So when our troponin binds to the calcium, right, it's going to undergo a conformational change, which is going to induce a conformational change in our tropomyosin, which is then going to change it. So now look at these binding sites. This is a myosin binding site that's exposed. Now this head group of myosin can bind onto one of these sites and undergo the ratcheting movement, right? So that's what's going to initiate our muscle contraction. Okay, so it's all dependent upon conformational changes in these proteins. Now, you've got that. So let's look at the whole thing. We still haven't kind of talked about why ATP is important. So here's our beginning step, right? We have our myosin. Our myosin is bound to ADP and it's bound to an inorganic phosphate. So this is like our initial conditions. Our tropomyosin is covering these binding sites for myosin on actin so that nothing can happen. We introduce calcium into the system. Calcium binds to our troponin. Troponin undergoes a conformational change. Now our tropomyosin undergoes a conformational change. Now myosin can bind. So myosin binds, that's great. The myosin is going to, once that myosin binds, it's the binding that basically pushes out this inorganic phosphate. So the inorganic phosphate gets released, and now this myosin only has an ADP attached. It has no inorganic phosphate. So when that inorganic phosphate is released, it induces what's called the power stroke or the ratchet, right? Because if this is the head of myosin, myosin grabs onto the actin, and then in a power stroke, it pulls it. And I'm going to show you the little animation of that. And so it actually pulls the actin filament 70 angstroms towards the center of the sarcomere. And so it's this movement that causes muscle contraction. So if all of the myosins latch on and contract at the same time in all of the sarcomeres, in all of the myofibrils, and all of the myofilaments, and every single muscle cell inside of your muscle, you're gonna have a muscle contraction, right? Okay, so but that's only one ratchet. You got to think you're going to have multiple ratchets in order to get enough of a compression of your sarcomere for you to see an effect on like your arm. So what's going to happen next is once that ratchet movement occurs, the ADP is going to be released, and now you're going to have an empty nucleotide binding pocket. Once that binding pocket is empty, it's ready to pick up another ATP. When ATP binds in the pocket, that is going to induce release of the myosin from the actin filament. Once that release occurs in the presence of water, you're going to hydrolyze that ATP into ADP plus PI. And look at this, we're back at step number 1. So this is how it'll continue contracting. It'll just continue contracting. Now, at some point you want to stop. At some point you want to stop contracting your muscle, right? I want to relax my muscle. So how do I stop contracting? Well, then I need a neuronal signal, right? What do I do when I want to stop? I need my neuronal signal to stop. So neuronal signal stops. What happens when the neuronal signal stops? Now you have to go back to the last section. Now what initiated this was that calcium influx. So how do you reverse that calcium influx? You're going to pump calcium from the cytosol, and I want to put it back. Where did it come from? It came from the sarcoplasmic reticulum. I'm going to run out of room. Reticulum. Y'all know what that is, sarcoplasmic reticulum, right? So now you're actually moving against the gradient because the sarcoplasmic reticulum has so much calcium in it, it couldn't dump out all of it. So you're trying to push calcium back into the sarcoplasmic reticulum against the gradient. So what kind of a pump are you going to use? That is your primary active transport protein. And if you remember which one it was Circa. CA, yeah, right? So now you have no calcium. So your myosin binding sites on the actin are now blocked again by tropomyosin. And then the thick and thin filaments, right, they're kind of overlap, are gonna slide past each other and stop at their resting state. So that is muscle contraction. So I'm going to post a separate video that has the animation for you. So have a great rest of your day.